Thank you, Jane. Our next speaker is Dr. Noelle, one of our postdoc alumni. Noelle is affiliated with 5 PhD programs: BMS, Tetrad, Neuroscience, Chemistry and Chemical Biology, and Oral and Craniofacial Sciences. Her lab, within the School of Dentistry at UCSF, studies the cell's biological processes that promote and maintain sensory signaling and neuronal plasticity through development in the tiny worm called C. elegans. Noelle.
Thank you, Liz. So I'd like to talk to you today about you. Yeah, you, Liz. I'm really interested in what made you you. And we've heard from Mike that you have all this DNA, but does that make you you? If you had an identical twin, would she be you? I don't think so. What really intrigues me is how your DNA, how those cells that DNA makes, has intersected with your education. And many of you have gone through UCSF. All the rigorous training that you have gone through has changed your cells, your circuits. And what I'm really interested in is understanding how those tiny circuits that we think are linked up to create a beautiful brain is regulated by its experience. And Liz is complex. She has 10 to the 11th neurons in her brain, with 10 to the 15th synapses or connections between these neurons. And that blows my mind. It's like the stars in the sky, the number of synapses or connections these neurons could make with each other. So to look at things at a simple level, yet also the building blocks, the Legos, if you want to say, for her brain, I chose to join Corey Bargman's lab when she was here at UCSF, and she might be known to you as the leader of the Brain Initiative that Obama started, and now the president of Chan Zuckerberg Initiative. But anyway, when she was at UCSF, I joined her lab as a postdoc to study this tiny organism, C. elegans. And they're up here in gray, and they're moving about. They're those little dust specks that are moving on each of these gray panels. They're 1 millimeter long by 0.1 millimeter wide, and they're completely transparent. And the beauty of a transparent organism is that you can look at them through a microscope, a tiny transparent organism. And so the bottom panel here is where I show the head of a worm. Now, what we've been able to do, which really is very important for our study of these animals, is we'll be able to look at each and every single neuron within the head of the worm. They have only 302 neurons with 1 million connections between them. And this reduces the complexity greatly. And so what we were able to do is we grabbed this protein from jellyfish. It's called green fluorescence protein. We hooked it up to another protein that binds to calcium. And when the calcium is bound, then the green fluorescence protein shines. And it's these yellow blobs we see are neurons that are expressing the green fluorescence protein bound to the calcium reporter. And calcium, if you remember, is actually the signal by which neurons speak to each other. The more calcium, the louder they're shouting. So this neuron here that you see in bright yellow is shouting to its neighbors. By contrast, those neurons that are kind of dark purple, you can't really even see them, those guys are quiet. Okay, so we were able to look at the brain of the worm. But what does this do for us in terms of learning and memory and plasticity? Because that's what we're really interested in. So these worms can also learn. And the paradigm is shown up here. Basically, what I've done is I have a bunch of worms here, and they're really hungry, and I've placed this odor where my pointer is, and this odor, they love it, right? Look at them. Oh my gosh. They're like graduate students going to free pizza, and they can't get enough. So, but they're not stupid. I give them this odor, and I starve them while I'm exposing them to that odor. And I do this for about an hour. And then I challenge them again. And I have the odor here at this red pointer on the right side here. And if you look at the way these specks are moving, they're kind of not that interested. They've realized that this odor is really not going to lead to pizza. Instead, it's nothing. And if I keep training them with cycles of this training, they begin to actually hate the odor and run away from it. And so this is plasticity, and we can achieve this change in plasticity, and they keep that memory for a day. And okay, the lifespan of a worm is about 2 weeks, so that's a long time, 1/14 of their life. They've learned that this odor means nothing to them. So The next step then is to take these worms that are naive and put them in a device so that I can look at them under the microscope, like I told you. And we got together with my colleagues that are amazing collaborators at UCSF, Torsten Whitman, a microscopist, Saul Kato, who's an amazing computational biologist. And if you could run the slide, the movie, we watched this worm's brain. You can see it moving here as odor was being flown across it. So what we have here is a very simple circuit, and I can identify, because I spent too many years looking at these, every single neuron in this 100 neuron ensemble, and I know which neurons are those neurons that are sensing the odor. I know which neurons are those that are causing the animal to move away or towards the odor. And I also know the neurons that are in between these. So what I've just described to you is a very simple circuit, the circuit that I'm interested in Liz's brain. You have an input, you have an output, and you have in between. These are called interneurons, and that's where the interesting stuff happens, the interneurons. And so what I'm interested in understanding is how information flows into this tiny circuit, and what happens in the interneurons. And so what do we do, and how this changes with training. So what have we learned when we stuck our worms into these microscopes? Well, one thing my postdoc Chantal Brufman learned was that the signal coming in to the sensory neurons, the input signal to the circuit, is very, very robust and very, very faithful. If there's a stimulus, it lights up. It talks very clearly. What we also found, and this is actually a lot of work from other labs as well, found that the motor neurons are very faithful. They fire, and the animal goes where those neurons are supposed to take it to go, okay? But in between the sensory neurons and the motor neurons lay the mystery. And this is what was really cool that we hadn't anticipated. The signal becomes completely degraded. This is not like computing. The signal takes on variability. And this variability in the signal is key to learning. We found a second thing when we looked at these animals. I didn't tell you this, but in order to learn, these guys have to fall asleep. So, I didn't tell you this, but after 3 cycles of training, these animals are tired, and they lay on the plate, and they don't do anything. You know, they take like little 30-minute naps. And if I disrupt their 30-minute nap, they never learn, okay? This plasticity does not occur. So, what I'm poised to do now, and I'm starting to do this, is getting together with Saul again, and we're putting those sleeping worms. But these sleeping worms are learning something. And I've been able to find sleeping worms that have learned that this odor is bad and watched their brains. And what do you think is happening? Just that, nothing. The neurons are quiet. They're doing something, though, right? Because the memories are accumulating. And the mystery now, to me, is what's going on? There's something going on. It's quiet. And what can we learn from this very simple circuit? Well, I think we can learn a few things. One is how biology, evolution, designed a system that computes whether a stimulus is good or bad, allows an organism to survive in the wild, which means it's integrating many more stimuli than I am actually giving it in the lab, and coming out with a ethologically relevant behavior. And so this evolution, I believe we could mine to create computers, perhaps, that have computing that is biologically based, more flexible, perhaps self-healing, and maybe part of those second-generation prosthetics that could help us as we blow through our own circuits. Thank you.